Hello, my today's report will focus on invasive diagnostics of musculoskeletal disorders. Today there are quite a lot of different methods for skin microcirculation analysis. There are laser speckle contrast imaging, methods based on optical coherence tomography, photoplatismography, and laser Doppler flowmetry. In general, the presented methods are already used in clinical practice. However, only photoplatismography and laser Doppler flowmetry are the most widely implemented methods due to relatively simplicity and good methodological support developed by scientific community. Laser Doppler flowmetry methods is based on the registration of laser radiation scattered inside skin by moving red blood cells. After that, power spectrum of registered photocurrent signal is summed by certain bandwidths of Doppler broadening frequencies. Then, power spectrum is normalized by DC part of registered photocurrent. We suppose that analysis of power spectrum distribution can provide a useful information about blood microcirculation and can be applied to existing laser Doppler flowmetry methodology of various diseases diagnosis. So, the main aim of our work is to develop the new approach to signal processing in laser Doppler flowmetry that provide new feature space for diagnosis of diabetes mellitus type 2 vascular complications. Our main suggestion is to utilize the amplitude distribution by the spectrum. In this research, we split the spectrum into 64 subbands from 0 to 12,800 Hz by 200 Hz per bandwidth and process the blood perfusion parameter from all subbands. Thus, we receive a blood perfusion distribution by Doppler broadening spectrum. To test our approach for diabetes mellitus type 2 complications, we developed the following protocol. We chose the dorsal surface of foot near the tooth as the area of interest and placed the Doppler probe coaxially combined with Peltier element. This area was chosen because the DM2 complications are usually connected with microcirculation of foot skin. Lack of blood perfusion leads to loss of proprioception and sense of pain, as well as causes ulcers and even necrosis. Experiment consisted of 3 stages. At the first stage, a basic level of blood perfusion was recorded under 33 Celsius degree temperature. Then the temperature was gradually increased up to 42 degrees for 5 minutes by 2 degrees per minute. Finally, at the third stage, the perfusion was registered for 20 minutes. The temperature of 42 degrees was chosen because it provoked the axonal reflex, an increase in skin blood perfusion when this temperature is reached. Here is the table with quantity of volunteers and patients with diabetes mellitus type 2 involved in this study. Thus, the main idea is to extend the well-known methods of the blood perfusion analysis with evaluating the distribution of perfusion over the frequency of laser radiation Doppler broadening. Our idea allows us to evaluate changes in amplitude distribution by frequencies. At the first step, registered spectrum is normalized by its own sum. Thus, we can evaluate what part of the signal is concentrated in different subbranches. The next step is computing the cumulative sum chart. This chart provides the ability to find how much signal is placed at a certain frequency. Finally, we calculate a mean cumulative sum chart across the selected time range. The following signal time bands were selected for the proposed analysis. The letter A means averaging by full time duration of stage 1. The letter B means averaging by axonal reflex period. By B-A, blood perfusion parameter describe a reserved features of blood flow. As a matrix of spectrum broadening, we chose area between curves parameter averaged by stage 1 and axonal reflex period respectively. Here it is simply recognized that signal is redistributed in high frequencies. The statistics comparison of selected parameters between 3 groups are shown on this slide. Significance of differences was tested by my-Whitney test. Analysis of B-A blood perfusion parameter demonstrated differences in groups of volunteers and patients. Evaluation of the area between curves parameter showed the differences between young volunteers and patients. Further, we made a linear discriminant analysis between groups of volunteers and group 2 versus patients. It should be highlighted that the same increase in diffusion from the base level to the axon reflex can be described by rather different distribution of amplitudes over frequencies. At this slide, ROC curves and area under curve parameter evaluation are shown. Here we compare ROC curves of individual parameters with linear discrimination function of parameters, both in two samples, as described in the previous slide. Area under curve parameter for LDA between groups of volunteers was more than an area under curve for detached parameters. Thus, high area between curve parameter means that overall blood perfusion adds up from red blood cells with higher velocities compared with cases of low area between curve parameter despite of similar by-a blood perfusion increase, and the area between curve parameter can be distinguished feature for discrimination David mellitus type 2 complications. Here there are the conclusions for this study. In the future we suppose to add new experiments in 3 groups to improve our metrics and linear discriminational analysis. We would like to thank the Russian Foundation for Basic Research, Russian Science Foundation and Academy of Finland for the financial support, as well as the healthy volunteers and patients who participated in this study. We would like to see your questions in the form below. Thank you for your attention.